Wonderful. Section 12-4 is actually a section a lot of people find very interesting, and that's because it deals with how alterations in chromosome number and structure can cause some changes and result in genetic disorders. So as you can imagine, our cells depend on the number of chromosomes that we have and the arrangement of those chromosomes. We now know many genetic diseases are caused by changes on the chromosomes. So what happens when we have large-scale changes to chromosomes besides simply diseases that are carried by certain alleles? Well, what ends up happening is we can end up with very challenging phenotypes and disorders that result. Most of the time, these large-scale changes lead to early spontaneous abortions or miscarriages. They don't actually make it out of the womb, or if they do, they can cause a variety of developmental disorders. We have observed, and don't quite know why yet, that plants tolerate genetic changes much better than animals do. And typically, these changes in meiosis cause a much bigger problem in other animals compared to plants. So one of the biggest ways that we get changes in our organisms is by abnormal chromosome number. So in non-disjunction, as you can imagine based on the name, this means that pairs of homologous chromosomes do not separate normally during meiosis 1. So typically what happens is one gamete will receive two of the same type of chromosome and another gamete receives no copies. So you might see one gamete gets 2 copies of chromosome 21 and the other one gets 0. And as you can imagine, when they fuse with another gamete, that can result in an abnormal chromosome number. So with non-disjunction, this is one example of how it could happen. We see in meiosis 1, typically those homologous chromosomes will separate 2 on each side. In non-disjunction, these homologous pairs do not separate. So we get 4 pairs of chromosomes on this one side. And then in meiosis 2, so that could be one way non-disjunction could happen. Or we could get non-disjunction in meiosis 2, where we see that the chromatids are not separating properly. And in either case, we end up getting gametes that either have too many or too few chromosomes. We call fertilization involving gametes where non-disjunction occurred aneuploidy. And typically, offspring who are aneuploids have an abnormal number of a particular chromosome. And we usually break it down as one of 2 types. Monosomic zygotes have only 1 copy of a particular chromosome. So that would be, for example, a gamete that fused with a gamete that didn't have any copies of that chromosome. Or we have a trisomic zygote, which has 3 copies of a particular chromosome. Super helpful. Polyploidy is a condition in which an organism has more than 2 sets of complete chromosomes. So there are some examples there, and this is pretty common in plants, but not in animals. So animals typically don't express polyploidy, but some plants do, and they tend to be relatively normal in appearance. The other type of changes to chromosomes that we can observe that cause large-scale disorders is involving the breakage of chromosomes, which in turn alter their structure. So chromosomes can have segments deleted, they can have segments duplicated, they can have segments inverted, in which there is one chromosome section that reattaches to the original chromosome but in the reverse orientation, so it kind of flips. And then we have translocation, which is when one segment of a chromosome breaks off and then that fragment might join another chromosome. So for example, deletion, you can see part of this chromosomal segment is removed. Duplication, you can see this segment was repeated. Inversion happens when the segment, maybe order, gets reversed. And then translocation is where you have multiple chromosomes and one section of one gets moved to another, and usually the opposite will happen. And since these aren't homologous pairs, it's not a synonymous switch. When we take a look at these disorders, Deletions and duplications will occur frequently in meiosis due to crossing over, and duplications and translocations tend to be quite harmful. And large-scale deletions can also be lethal. So these aren't great things to happen to your chromosomes. And this is often why we see miscarriages occur. A diploid embryo that is homozygous for a large deletion, so for example, if both of their chromosomes had large deletions, they're probably missing a number of essential genes and they will typically die before they come to term. And like I said, the duplications and translocations tend to be pretty harmful. In inversions, as long as the balance of genes is normal, they can be okay, but if the expression of the genes is altered, then that may influence the phenotype. So we know of several human disorders that are due to chromosomal alterations, either whether they have the wrong number of chromosomes or they have an altered structure of chromosomes. Some of the types of aneuploidy, like we mentioned, upset the genetic balance less than others, and those individuals are able to actually be born and survive even beyond birth. But typically, they will express a set of symptoms, and we term this a syndrome, and those syndromes are pretty characteristic of the types of aneuploidy that we observe. So one of the most common syndromes we know of is known as Down syndrome, also known as trisomy 21. And this is because individuals with Down syndrome have 3 copies of chromosome 21. So you see the trisomy over here. It is unknown why at the moment, but we do know that the frequency of Down syndrome increases with the increasing age of the mother. So the older the mother is, the more likely it is that she will have a child with Down syndrome. So you can see this is an example of the karyotype of an individual with Down syndrome. You can see they have 2 pairs of all the chromosomes, including their sex chromosomes, but she has 3 copies of chromosome number 21. And this is kind of an expression of some of the physical manifestations you will see of the symptoms associated with Down syndrome. There is also a pretty common occurrence of aneuploidy of sex chromosomes, and we think that this is true because this non-disjunction of sex chromosomes produces a lot of conditions, but for the most part, because the Y chromosome contains very few genes, these syndromes affect the genetic balance a little bit less than others, and so these syndromes are a little bit more common than other syndromes you might see. So Klinefelter syndrome is the result of an extra chromosome in a male. So they would get, for example, this would be a male with XXY. And these men tend to develop male sex organs, but their testes are very small, and the man is typically sterile. And men with Klinefelter syndrome typically express lower intelligence. About 1 in every 1,000 males is born with an extra Y chromosome, XYY, and we don't observe any physical changes to these men as of yet, so they typically are not characterized with a syndrome. And in a similar vein, females with trisomy X, who have XXX, have no unusual features except being slightly taller than average. However, with Monsonomy X, where a woman only has 1 copy of X, which we would refer to as X0, they express a syndrome known as Turner syndrome. They're phenotypically female, so they appear to be female, but their sex organs never mature, and so they remain sterile. And for the most part, we consider these women to be of normal intelligence. And it's also, Turner syndrome is the only known viable monosomy in humans. So it's the only example of monosomy that we know of where the individual can live till birth and live a life. Then we can talk about a few disorders that are caused by structurally altered chromosomes. So this would be when we see inversions, deletions, duplications, and translocations of chromosomes. So there's a syndrome known as chat or cry of the cat, and this results from a very specific deletion on chromosome 5. Individuals born with this syndrome are severely intellectually disabled, and when they cry, it kind of sounds like a cat meowing. So that is where the name comes from. And these individuals typically die in infancy or childhood. But remember, this doesn't really affect the occurrence rates of the disease because it has to do with the actual chromosome structure themselves, not any genes that are passed on, like with some of the other diseases we've discussed. There's also certain cancers, such as chronic myelogenous leukemia, which are caused by translocations of chromosomes. So this particular form of cancer is caused by a reciprocal translocation of pre-white blood cells. So a large portion of chromosome 22, which you can see is already pretty small, exchanged with the tip of chromosome 9, and that translocated chromosome 22 is often referred to as the Philadelphia chromosome, and this creates a new fused gene that tends to lead to out of control cell division of the white blood cells. So that's what ends up causing the cancer in this case. And that is just a few examples of some of the different types of genetic disorders that can result from alterations in chromosomes.